Cheryl Kerfeld is the Hanna Distinguished Professor of Structural Bioengineering in the Department of Energy Plant Research Laboratory and the Department of Biochemistry and Molecular Biology at Michigan State University. She also holds appointments with the Physical Biosciences Division of Lawrence Berkeley National Laboratory and the Berkeley Synthetic Biology Institute. Her research group focuses on structure-based characterization and engineering of photoprotection and the carbon concentrating mechanism in cyanobacteria. Her group is also developing bacterial microcompartmental-based systems for metabolic engineering. She's going to explain all of that to you in the next 10 minutes. The Kerfeld group combines methods in bioinformatics, cellular imaging, synthetic and structural biology for the engineering of bacterial metabolism. Cheryl received her bachelor's degree in biology and English at the University of Minnesota. She has a master's in English literature from the University of Minnesota via the Regents Scholars Program, and she has a PhD in biology from UCLA. Our recruitment of Cheryl was quite challenging because it's always hard to pry someone away from sunny Berkeley, California, but we managed to do it, and I'm very happy we did it, and I want to welcome Cheryl to our meeting today.
All right. Thank you very much. Good morning. All right, and so what I'd like to tell you about today is one part of the research that goes on in my group, and it's related to improving photosynthesis. And the implications of this are, of course, many. So one can think of, okay, this is really important for crop productivity, and with a changing climate and so on. And also, there's new developments now to turn bacterial cells that do photosynthesis into little microbial factories for producing things that we previously used to derive from petroleum, right? So there's a real push for this kind of metabolic engineering, and that's some of the work that we're doing. And so in order to do this kind of thing, and what you're looking at here are big fields full of ponds full of algae that are producing different types of products, commodity products. In the laboratory, we deal with these kinds of organisms on quite a different scale. And what you're looking at here is bottles of cyanobacteria, and these are photosynthetic bacteria, and they are actually the ancestor of the chloroplast. So a lot of the things that we learn in cyanobacteria we can apply to plant research as well. And here you're looking through a microscope at one of these cells, and now you're looking through an electron microscope at a photosynthetic cell. And what we study, actually, breaking this down, so here's our photosynthetic cell somewhat simplified here, is the way that we break this down is part of my group studies how we capture light energy and turn that into chemical energy to make sugar out of carbon dioxide. Part of my group is involved in engineering the transporters for the raw materials that we're going to use in our microbial cell factories. And here's an example of the factory itself, right? So these little oddly shaped compartments that we find in bacterial cells. And I'll tell you, up until about a decade ago, people didn't appreciate that bacteria actually made these kinds of structures. And I'm going to focus on telling you a little bit about what we've learned about this architecture today so that you can sort of see where we're going with trying to re-engineer this to produce some high-value compounds of interest to us. So this is how we have to approach this kind of thing. We want to, in my group, a particular emphasis is understanding what things look like. So it's kind of like if you thought about having to fix your car without being able to see the different parts, the different machinery, it'd be a very difficult task. So instead what we do is we purify the individual compartments, and again you're looking through an electron microscope here, and this is about 200 nanometers across, so very tiny. And then what we can do is we can take that purified compartment, and we have special tricks in the laboratory to separate it into its different component pieces, sort of like the spark plugs, the carburetor, various different parts. But then you see what we have is just a parts list. We don't really know what these individual pieces look like. And that's only so useful, okay? What we'd like to be able to do is understand what these things look like, how they snap together, and then how they function in the cell. So this is how we do this particularly in my lab, is again we start with our bacterial culture collection, our culture condition, and we purify maybe one protein out of this, and we coax it into forming a crystal. And this is a really non-trivial undertaking. And what you're looking at here is, again, you're looking through a microscope, but what you're looking at here is maybe 1/3 of a millimeter across. So it's a teeny tiny crystal that we use. Now what we do is we take these to California, where we have an X-ray source that we use to shoot at the crystal, and we obtain a pattern like this. But obviously it's not exactly a direct and easy thing, because there's lots of hidden ambiguities in this that we have to sort out. And actually the data looks something like this. And so I won't go into the details of that, but that's basically how we solve a structure to learn what a protein looks like. And from those kinds of studies, my group learned that the basic building block of something like this in the cell are proteins that are hexagonal and proteins that are pentameric, so 5 sides and 6 sides. And we also learned that the hexagonal ones tile together, just as you see right here, kind of like the tiles in the bathroom on the bathroom floor or something like that. So that was really interesting, and that's how we think the facets or the sides of these kinds of compartments are made. And then if you think about icosahedral geometry, okay, many icosahedra are built up of hexamers and pentamers. So what we can do is we can take and we can just snap out one of the hexamers and we can bend it a little bit, and we can actually fit a pentamer into this. And this is actually the same sort of underlying architecture that one has in a soccer ball. Look at it sometime. You'll see that soccer ball has 12 pentameric pieces in it and a bunch of different hexamers. And so that's actually the underlying structure of these kinds of compartments. So functionally then, we have the building blocks for the outside, the shell. Okay, so these are like the walls of the factory that have to be selectively permeable so we can get things in and out. And we work on ways of controlling that permeability. And in the inside, we have the enzymatic or the catalytic machinery. So if we want to build isoprene or we want to make some sort of compound here, we set this up in the inside. So what I've shown you so far is what we can learn from understanding the structures of the individual building blocks. But really the question is, how does this all come together? What's the roadmap or the instruction set for putting something like this together? And so again, the enzymatic machinery, the walls, and even a central question for us was, okay, does the shell assemble first and then we put things in, or do we build things first and then put a shell around it? Okay, and there's examples in biology of both of those sorts of strategies. So the next thing that we did is we took one of those shell proteins here, that's part of our compartment, and we labeled it with a fluorescent dye, right? And now you're looking at a field of cells here, and each one of these bright dots, is the fluorescently labeled factory, okay? So then the next step and what you can see then a little bit better here, each one of these green dots is a factory inside of a cell. Each one of these is an individual photosynthetic cell. Okay, so now we learned that we could actually label our factories. That opened up the doorway for us to be able to understand or to follow how they assemble in real time. And the way we do that is we set up the conditions before a factory is built. And we've got some of the components labeled with fluorescent dyes. Okay, and that's what you're looking at here. Each one of these red things is a cell, okay, and you can see there's kind of a diffuse blue color throughout all those. That's our labeled protein. And then what we can do is we can turn on the conditions for building the factory, and we can follow that the proteins come together. And this happens to be one of the internal proteins that's labeled here. And you're watching this through a microscope now, a special kind of microscope that allows you to see fluorescence. And what you saw was just all of these scattered building blocks coming together, forming these puncta, and then later on there's actually a sequel to this movie that I'm not going to have time to show, but later in the sequel you see them get distributed across the cell. Okay, so this is a key finding for us because it tells us that these sorts of factories are assembled by putting the building blocks or the catalytic machinery together first and then putting the walls around it. That opens up the door for us to put whatever kind of catalytic machinery we want to in the interior. So maybe we can make precursors for rubber, for example, in something like this. So that's the general approach. So now we know how these things come together, and what do we do with them next? Well, one example is we can try to take this sort of naturally occurring compartment that's found in bacteria but not found in plants, and we can actually install it into plants for doing something desirable that we might like to do. And then a particular area of focus of my group, and this is a cover article that we had a few months ago in Trends in Microbiology, so this is like the big time, right, when you're on the cover of a journal like this. So our particular interest is engineering microbes for these kinds of things. So that's where we're going. And I'd like to just tell you a little bit about the people that did this. This is my group, and some of them are in California, but the majority of these folks are here at Michigan State. And I'm really grateful to my colleagues in the MSC, or the DOE MSU Plant Research Laboratory, because we're really interested in photosynthesis. I'm also part of the Department of Biochemistry, and I'm a Hanna professor, and I'm grateful for the opportunities and the resources and to be here at Michigan State. Thank you.
Questions, comments, questions? Would you, it help people understand sort of if knowing all of this and the next steps in your research, and if we were having this conversation 3 or 5 years from now, what kind of difference could they see in the world from this?
Thanks. That's a great question, because one way of really motivating young people, and young scientists are really motivated to do something positive like this. So, for example, trying to make something that's normally derived from petroleum, but make it in a green chemistry sort of way. And that's what we're interested in doing here. Also, we have the corollary benefit of some of these factories that we find in bacteria actually use carbon dioxide as a raw material. And so that has the potential then for perhaps contributing to mitigating some of the problems that we think we're experiencing from that. So
that's the big, I mean, it's
Climate change.
it's part of the question of how do you think about plants as a positive effect on climate change, as well as, you know, alternatives to petroleum and other kinds of things. It's really, but it's fascinating, very fundamental work, very, very difficult, but has this sort of downstream potential. We're so pleased that you're at Michigan State with our group and having an opportunity to be a part of what we think is a pretty special group of people, but you've made it even more special.
thank you.
Thank you.
Thank you.
Thank you.